everyone. Welcome to Cure Talks. I'm Priya Menon, and today we're discussing UltraCAR-T cell therapy and explore the scope and possibilities of using this therapy in management of multiple myeloma. Joining me on Cure Talks today is a myeloma panel of Gary Peterson, Jack Aiello, and Yelik Biru. The featured expert we have with us is Dr. David Sallman from the Department of Malignant Hematology, Moffitt Cancer Center. Dr. Sallman's research interests focus on the development of novel targeted therapeutic strategies for patients with myelodysplastic syndromes and acute myeloid leukemia. Welcome to Cure Talks, Dr. Sallman. Thank you for taking this time to join us today.
Sure, thank you for the introduction.
We have a lot of questions to address. In fact, we have a lot of audience questions too, and I'm going to just jump right in. Dr. Sallman, we spoke to Dr. Carl June on this platform back in 2017, and there has been a lot of research on CAR-T therapy and the use in cancer since then. Can you just start with what is UltraCAR-T cell therapy and how is it different from the traditional CAR-T cell therapy?
Yeah, so maybe just to take a quick step back, just to set the basis for what we're trying to treat. So for B-cell lymphomas, multiple myeloma, of course, CAR-T has really been a paradigm shifting therapy for patients that have relapsed refractory disease. I think similarly, and I think even of higher concern in patients that have acute myeloid leukemias, we have very limited treatment options once they sort of fail standard therapy. And for elderly AML, as an example, where hypomethylating agent with venetoclax is the standard, you still have a median survival of 14 months, and essentially all patients ultimately relapse. And if you don't have a targetable mutation, which is really only about 30% may have a targetable mutation, then there's actually no approved standard therapy or even therapy that's near approval in that setting. So we're really hoping that we can have sort of a novel CAR-T approach to give our patients, hope, both in the elderly setting, which I just mentioned, but still many younger patients ultimately fail standard of care therapies. And the challenge is when we're saying how poor some of these patients do, we're talking a median overall survival of less than 6 months across the board when we have a relapse refractory AML patient that has sort of failed standard of care treatments. And so one of the challenges with traditional CAR-T therapy can be the processing time. So 3 to 4 week sort of time period when your patients already have a survival very short-lived and have lots of issues related to their cytopenias, such as infectious complications and others. Trying to have an approach that can sort of give the therapy at the right time is important. So what UltraCAR-T is, it actually allows for an autologous therapy. So we're still using patient cells, but we can basically go from taking the cells out or apheresis and infusion into patient within 2 days. So it is sort of rapid, as you could hope for from that perspective. We presented sort of our trial and progress data at ASH, but what's nice is we've not had any manufacturing, failure issues, and we're continuing to go on to dose escalation. So I think the ability to rapidly dose patients with this platform is here. And again, this is allowing us to use an autologous platform. I know I'm a little bit long-winded and I'll take a break in a second, but I think the other main approach to sort of rapid dosing of patients is sort of using somebody else's cells or an allogeneic or off-the-shelf approach. The challenges that can be there is that patients may reject these cells quite rapidly. And so there may be issues with persistence and expansion, which ultimately correlates with efficacy, at least in the B-cell, both in ALL and B-cell lymphoma. And so potentially having a rapid but autologous product may be able to give you both the quick turnaround time, but ideally potentially better issues from an expansion and persistence perspective.
Thank you, Dr. Sallman. I think I'm going to let Gary start and lead this panel discussion now. Gary, all yours.
Okay, well, thank you, Priya, and Dr. Sallman, thank you so much for giving us your time on this. We're all very much interested in CAR-T. It's pretty much taken or center stage for all treatments in the B19, which Dr. June had championed, as well as now going to BCMA and being used in the myeloma setting. And for you, AML, which really doesn't have many treatment options available. In the past, all new drugs that we had in AML anyway, have had, and in myeloma and other blood cancers as well, have had overall survival and progression-free survival that were in months and not and weeks. And as a result, there wasn't any breakthroughs at all. But in CAR-T therapy, they've been, those have gone, progression-free survival and overall survival have gone all the way to, many months and now years for both. And as a result, it's very exciting. And given what Carl June had said during that program that Priya had just mentioned, she had mentioned that, what he mentioned was during that time, that CAR-T he thought was going to be a cure, and that it was just a matter of finding the right target or the right combination of targets, and that cure was where he thought the whole thing was going to be. So right now that's great information. That's nice to know. But there's, even with those great successes, they include a lot of challenges like high cost. They're talking $500,000 insurance coverage. I don't know. The insurance seems to balk at every time we patients want to try to get something paid for. And if you put a $500,000 price tag on it, that's even worse. And finally, not finally, but also takes, like you said, 3 to 6 weeks to manufacture and has side effects like cytokine release syndrome. Now you're tackling AML. And like you said, there is very few treatments for refractory and relapsed AML. And as a matter of fact, life expectancy is in just a few months when you get to that point for AML. So I applaud you in all that you do to take on such a very difficult subject. And I think you're frankly quite amazing. And thank you for that. Probably the most important question I have for you, and that is, does UltraCAR-T work? And is this treatment have efficacy? And finally, is it durable? And how are your results to date?
Yeah, so great questions. I wish I had excellent answers to all of them. I think, everybody looks to the B cell, the CD19 and the BCMA positive diseases. Those are really optimal targets. I think the challenge in AML is we already know there will never be a perfect single antigen optimal target, meaning there's not one marker that's expressed on all leukemia stem cells or bulk blasts that we can target. If we had that, we would already be as far along as where they're at in CD19 positive disease. We have a good sense of markers that are worthwhile to target. The two kind of leading candidates of targeting right now have been CD33 and CD123. The pros is they are expressed on a high percentage of bulk blast with maybe a little bit more heterogeneous expression on leukemic stem cells. But again, they're not uniformly expressed. One negative of targeting them is they do have some expression on normal stem cells. And that's really right now in concepts that are targeting 33 or 123, really these patients are required to have a backup allogeneic stem cell transplant donor, because at least in mice modeling, a lot of this work was done at Sargill's group, but a couple of other labs have shown it similarly, is that you could induce aplasia or sort of the lack of ability to produce blood again. So that somewhat limits our current, clinical trial patient. Whether or not that happens in patients is a little bit unknown. So I think there's kind of several lines of research going on that are important. One is, what is the actual activity when you target CD33 or CD123? The answer is, as of today, we don't know. I do think over the next 12 months or so maybe by ASH of next year, ideally with our trial and with several other trials, both targeting 33 and 123, we'll have the answer once we've gotten to our, through our dose escalation, what is the activity or not with targeting one of these antigens. I think, let's say we don't have activity, the ultimate issue may be you need to target more than one antigen. So for example, in China, there's a compound CAR targeting CD33 and CLL1, and they actually had quite a bit of activity in a limited cohort. They had 7 of 9 patients presented at EHA of last year that had sort of a MRD negative complete remission, and it appeared that those responses were durable. Small number in that trial has gone slowly, but again, that's targeting 2 antigens. So what is the benefit of targeting one? Again, we're early in our dose escalation. Our hope would be by, ideally, I would say over this next year, we finish it. So we got to get to the cell dose. We've not had manufacturing issues. We've not had safety issues in early dose levels. But again, the major question of what the activity is of these, we don't really know. And again, there are major safety issues that are unique in AML that are not present in patients that have multiple myeloma or a lot of the B cell malignancies. I think once you know the targets that are key, so if 33 is great, well then that's easy. We can rapidly go forward with the current clinical trial and work to try to get an approval of a therapy. If 33 is not enough and you have to target 33 in another marker, then I think what's important is, are your platforms that you're utilizing able to be rapidly adapted? So one nice thing with the sort of UltraCAR-T program, it's a non-viral vector-based system, so it uses a plasmid with the Sleeping Beauty system. It is rapidly changeable. So if we needed to target more than one marker, I think we would likely be able to easily do that. The challenge is once you do that, you have to kind of restart from scratch, your dose escalation, your safety, but at least you're utilizing relatively the same, same technology that from that perspective should not, create differences. So the problem is, as of right now, there are so many unanswered questions that I can't give a great single answer to your question. But my hope is that yes, these therapy and potentially with the UltraCAR-T platform will be sort of a novel cellular therapy option for patients in the future. But again, many of these questions are still unanswered.
So if it was available, would you be able to include CS1?
I mean, I think we have or sorry, go ahead.
Along with 33 or add 120.
Yeah, I think the sky is the limit. So I mean, what's nice if you target more than one antigen, you can create it 2 different ways. So one is you can try to create it from a safety perspective, meaning that the T cell will only kill if it sees both markers. So you could say, hey, I'm going to require it to be CD33 and CLL1. That may be very specific just to the leukemia cell and spare really any normal cells. And some of this work has been published over recent years where people have done large flow cytometry panels looking at different combinations. So from a safety perspective, that's great. The challenge is what happens if you have some of your cancer cells that only express one marker or don't express either marker. And so this sort of concept of antigen escape may be a major issue. From my perspective, I'm really more on the boat of trying to do anything to increase efficacy. Right now, we just need options for our patients, and we have come a long way with managing the side effect profile. So to me, maybe you create the CAR so where it activates if it sees either marker. Again, that may be a little bit more toxic because it can potentially hit cells that are not the cancer cells. But again, our major issue, I think, right now is efficacy. We know even with CD19 CAR, for example, is these cancer cells can sometimes lose their CD19 expression. And that's one reason why these patients, are progressing, later on in their time course. In AML, we have no idea because we're still learning what is our, safety and activity of targeting it. But yes, I think both of these main concepts, again, targeting 2 antigens for more of a safety perspective, or targeting 2 antigens to increase the chance of getting activity and potentially not allowing these cells to escape. I think both of these are really important concepts, but sometimes we're getting a little bit ahead of ourselves because again, what is the activity which is targeting one?
Well, patients kind of like you guys looking ahead of yourselves.
Exactly. And there's other, I mean, there's a couple systems right now where and again, some of this work is at Sargil and Dr. Mukherjee as well. There's a couple companies, there's Vor Biopharma, and then Sargil's been doing this work at UPenn, where you actually remove CD33 expression off the normal stem cells. So the concept would be is you kind of go forward with like an allo transplant. You take normal donor stem cells and you knock out CD33, and then you give the CD33 CAR. And so it can't hurt the normal cells and it can only kill the cancer cells. I think it's an interesting approach. Again, whether or not 33 is the answer complicates this because you may go through a lot of this without having the answer. And that sort of platform is not easily changeable, because if you needed to knock out multiple markers, then that's probably not going to be safe, or you're not going to have the ability to do that. But there are other concepts looking to try to improve safety. Again, what I do like about the UltraCAR-T approach, one is the rapid manufacturing. Two is it incorporates this membrane-bound IL-15, which really allows these cells, it kind of gives their own food support inside the patient. And so instead of having, again, to grow a higher cell number, outside of the patient, these cells will actually proliferate in the patient, and probably safer from that perspective as well. And then of course, three, which we sort of mentioned already, is sort of the ability to adapt it if we ultimately need to target more than one antigen. But if we end up, if we could have a 30% remission rate in this group, that would be an extreme home run. I don't think we necessarily have to shoot for the 70% response rates that have been seen in the sort of CD19 and BCMA world. I think we really just ideally start with something. Now, the higher, the better. And I think to your question in the beginning, the durability matters because patients are going through a lot. A lot of times they still require several weeks admission. With all CAR-Ts, you typically have to be close to the center a month afterwards. So we really want these responses to last for. What's nice also with this technology is that if it does bring up the ability to repeat dose, much easier. And so let's say we had a response, but then it lasted only 6 months or 12 months or something. Could we then just redose at the right time? And then I think one of the biggest concepts, and this is really where things are moving in the CD19 world, is treating patients at the right time. So you don't necessarily want 90% blast when you're giving a cellular therapy. I think one of the most intriguing data were from Dr. Park's New England Journal of Medicine in B-cell ALL. And really the durable outcomes were in patients that had less than 5% lymphoblast. And so treating the patient at the so-called MRD level may be really where these therapies are best utilized. And what's nice is those technologies are already commercially available. So we can sometimes detect down to 1 in a million cells if there's still leukemia. Maybe that's the time to give a CAR-T. It will definitely be safer. And can we eradicate that MRD with the goal of ideally for cure in our patients? I think that is the long hope. Again, we're in our infancy period though right now.
You said, and it's kind of a follow-up on something you said earlier, in that if you target two different antigens, that it has to have both of them before it will engage. Is that what you said?
That is one of the two approaches. So that approach is done more from a safety perspective, because if you require dual, it will really only be expressed on the leukemia cell. It's extremely unlikely to be expressed on normal cells. The challenge would be, though, that the tumor could potentially escape if it was negative for one or both of them.
And how about sequencing?
Yeah, I think that's a good question. I mean, the challenge is, right, to get the answer of any single antigen and whether or not it works or not takes us several years to answer that question often. And so, we want to ideally come up with new options for our patients, sooner. So yeah, sequencing is, and like, for example, they're doing that CD19 followed by CD22, or do you try to do them both? These things are being done in B-cell malignancies where there are more options. That's a great question. Is it better to target them all at one time or sequence them? But I think the challenge is, by the time you get each individual answer, like the field is going to be moving too rapidly. So I think that is likely not a challenge unless we had multiple approved therapies, and then that question will come back, oh, okay, could we sequence these? I think it's the same issue. I think until there's another, so not CD19, but another approved CAR-T targeting a different antigen, the question of sequencing versus doing it at the same time would still be an open question.
Okay, well, thank you so much. What you've done is you've kind of wiped out most all of my questions now, but I appreciate that. And, I guess one, just from our perspective, all those issues that I mentioned before, that come out, that UltraCAR-T system states that it's a low cost, next day infusion, hospital-based system, which can be reapplied with the same or different antigens as targets. This just sounds too good to be true. So do you think this is fact, or is this science fiction?
I think everything that you stated could be fact. I think there are so many unanswered questions to truly make it fact, but everything that you stated is possible. These are still highly complex therapies. And I think, yes, can be decentralized, meaning that each center of excellence, could potentially be utilizing this, but I don't think this will ever sort of roll out to the community. So I think the answer is potentially yes, but it's probably going to be very specific based on each center of excellence and not something that you can just do anywhere, just given the complexity of managing a very, very sick group of patients. But I think it's giving us a lot of different hopeful next steps for ideally coming up with a new therapy for our patients.
Well, thank you. And at this point, then I'd like to go on to the questions from Jack Iola. And Jack has been living with myeloma for like 25 years. So he's an anomaly. We who know him understand that he really is an anomaly. So Jack.
Yeah, thanks for that introduction, Gary. Dr. Sallman, it's really interesting to me. And I did, as you're aware, ask some myeloma related questions, but
Sure.
Let me kind of turn them into questions that still might benefit myeloma patients.
Sure, of course.
The most interesting thing to me is the speed of the manufacturing process. Is there any reason that the same process couldn't be used to target BCMA instead of CD33 or 123?
Yeah, I don't see any reason, why that could not be done. And so, yeah, could you use the same platform and incorporate BCMA? From a technological perspective, I don't see any challenge, with doing that.
So ide-cel, for example, was just approved for myeloma and requires an infusion of 450 million cells. Does your process generate the number of cells required within the 2 days, or do you depend on cell expansion in the patient to occur to get up to that required minimum dose, if you will?
Yeah, it's a good question. So we've dosed, now into sort of the 1 × 10⁶, although we do have a potential dose plan up to 1 × 10⁸. So we think actually most likely we could make the number of cells, required without requiring the actual proliferation, in the patient. But, you have to investigate that in each patient population and how relapse refractory, what's their baseline lymphocyte count, but potentially we could easily dose that number of cells, with just in that rapid turnaround process. But there would be some things to answer that question definitively in the setting of treating myeloma patients.
You mentioned that your patients require to have an allo donor available in case things don't go well. For myeloma, allos aren't done very much because myeloma patients have very poor outcomes just getting through the transplant.
Yep.
So does that mean there's a difference in terms of your procedure?
Yeah, so that wouldn't be a risk. So for example, if we did UltraCAR-T targeting BCMA, there would be no need for requiring a backup donor. The only reason we required is because the markers we're targeting in AML, unfortunately, are expressed on normal hematopoietic stem cells. So BCMA is not, and that's what's been, it's nothing to take away, but in B cells, plasma cells are still B cells. It's a biological difference that they're able to exploit. Unfortunately, in myeloid leukemias, there is no perfect marker that's really expressed, only on the cancer cell. Or what's nice, even if you target 19 or BCMA and you wipe out B cells, people from an immune system perspective have still appeared to do very well without severe complications from that. So it's really a target difference, which is why that's required. But if we were target, BCMA, there'd be no reason to require a backup donor.
Got it. I got to say, I think your platform is awfully important and creates some breakthrough for myeloma. They are trying to come up with AlloCAR-Ts off the shelf.
Yep.
But as you mentioned, there can be risks associated with that. And if we can get down minimize the time of a couple days to generate CAR T cells and reinfuse them, that just sounds like a bandwagon that all the designers should jump upon.
I think, there's a lot of off-the-shelf approaches, but again, if they're ultimately not having the level of efficacy similar to the, autologous products or made from the patients, then this may be a way to both do the rapid, manufacturing part, but still allowing the autologous and ability to not get rejected right away. What's being required in the off-the-shelf allo products is typically more intensive lymphodepletion. So for example, several technologies use alemtuzumab because the product's been modified to not have CD52, but that may increase risk of infections and complications just from the lymphodepletion. So that's not something that would be required with an autologous platform, just sort of the standard fludarabine cyclophosphamide that all patients are getting.
Thanks so very much. I look forward to following the progress of UltraCAR-T, and I'll turn it over to Yelak.
All right. Good morning. I want to go back to Gary's and Priya's CAR-T 101 question and ask, why does it take such a long time for traditional, if we can call them traditional, CAR-Ts to be engineered and infused back to the patient?
Yeah, so I think typically with the viral vector-based systems, once you have the transfection, you have to sort of grow the cells up to get to the level that's going to be efficacious, and so that takes a significant amount of time. Again, in that 3 to 4 week period, with this sort of transfection system using what's called an ultraparator, that's not required.
Dr. Sallman, you're seeing the persistence of CAR-T as has been seen. Are you seeing the persistence of CAR-T as has been seen by Dr. Nora, that's D-I-S-I-S, Diaz, in ovarian cancer, also using the UltraCAR-T therapy? And they're working, I guess that's at Seattle Cancer Alliance. Seattle Cancer Alliance. Are you seeing the same persistence of CAR-T?
Yeah. So the only data we have this patient we presented at ASH. At least at the 6-month time frame, we were still able to detect the PRGN copy. So we could say at least 6 months. We don't have follow-up longer. And then I think that was a patient that was actually without lymphodepleting chemotherapy, which to us was actually quite surprising to have some level of persistence out that far, and it's probably more related to the membrane-bound IL-15. So again, that's an N of 1, which we should never make any conclusion or definitive answer. But the answer is we've at least seen persistence in a patient at a relatively low dose level without flu-cy. So potentially, with future dose levels, especially when we get to let's say our expansion dose level will be, and likely with flu-cy should increase the ability for persistence and expansion, then we'll have that answer. But already the answer is at least yes and an N of 1.
Okay. Recent administration dose patients in cohort 1 of your study, which have been given lower doses. So a dose so that they can check safety and tolerability. Have they been redosed with the same dose or maybe increased dose?
Yeah, so far no patient has been redosed. The technology definitely gives the ability to do that. It really comes down to sort of a safety and sort of in discussion, with FDA and things. So the trials have to be written very specifically, each patient followed extremely closely to ensure that we're learning what we need to learn. And so in general, patients are not allowed redosing. I think how I see it is once we get to the dose that we think is both safe and ideally effective, and let's say a patient has a response, potentially you can go back to the FDA on a single patient, case and say, hey, I'd like to redose this patient. That has been done with other cellular therapies, or perhaps, once we know our dose, we may be able to build in criteria. So if a patient has no toxicity, has no evidence of persistent cells, could we then redose on a case-by-case basis? That has been done with some other therapies, including CAR-T trials that I've been involved with. But I mean, the answer is right now, we're too early to be able to do that. But I think realistically, once we get to an expansion dose, I think there will be possibilities. And from a technological perspective, easy enough to do.
Here's one I think you might have touched on a little bit. These AML patients have very short time, like 3 months to live. One of the patients who was dosed was enrolled directly from hospice, meaning they weren't given a lot of chance to spend much time alive, and was given days and weeks to live. It was reported that 7 months later, is the patient still alive? Are all the patients enrolled in all cohorts still alive? And how many were dosed?
Yeah, so yeah, a lot of that, just given that the trial is still in dose escalation, I can't speak specifically of, but I do think, we were excited just by that one case, because as you pointed out, typically a patient with relapsed AML, especially let's say a patient that's enrolled on hospice, has very, very limited amounts of time. And even though that patient did not have an objective response, the patient did have blast clearance and had a quality of life, during, that amount of time. But I do think, looking at quality of life, looking at the durability of these responses over time is really important. And even above just objective responses, can there be some clinical benefit gleaned, from these cases?
Well, that's kind of a Lazarus syndrome.
Yeah, I'd hope so.
Gaelic back.
Yeah, Gaelic back. Gaelic.
He's back.
What you go running off for?
I know, right? We still need
just when we needed you,
CAR-Ts.
you take off.
We need CAR-Ts for computers. So I don't know if this question was already asked, but if it is not, are there any concerns with quality of distributed manufacturing now that this is going to be either at the hospital or a nearby facility than a centralized place? Can you address that?
Yeah, so I think it's a really good question, whoever asked that, because obviously this is sort of a unique system. Everything has typically been centralized, a single manufacturing source. So I think those questions will have to be answered. I would say what's likely going to happen is, there's relatively a limited number of true centers of excellence for cellular therapy, and likely these type of therapies will be done there. But I don't see any reason, because all of this would go through sort of extreme, quality, quality runs before patients are dosed, that it should be similar. But I think there will have to be, before the therapy was approved, very definitive proof that there are not any technological differences among centers, but I don't really see why there would be. Again, I don't see this personally. It's not that we have 1,000 centers that are, then giving UltraCAR-T. But again, the centers of excellence, across the U.S. and the world, again, there's no reason why they couldn't, again, develop, this technology in-house, ensure with tech runs that everything's, meets all the, batch specifics, and then, we can prove that there's some level of uniformity from there.
One of the early, at least, side effects of CAR-T in the traditional CAR-Ts was cytokine release syndrome. Are the overall normal CAR-Ts?
Yeah, so far, I mean, our therapy has been well tolerated. We have seen cytokine release syndrome, but at least in our early dose levels, have not had any dose-limiting toxicities or severe or refractory cytokine release syndrome. I think we benefit in the setting that we've learned so much from all of the investigators who've taken care of the B cell lymphomas and leukemias and myeloma. So we really know how to. We now give TOCI much lower thresholds than we used to, steroids when we need to. We've not really appeared, and I would say really in my myeloid experience, neurotoxicity has appeared to be significantly less. So is there something unique or different, or maybe we're just not far enough along in our dose escalation? But we clearly have seen cytokine release syndrome in CAR-T therapies, but to my knowledge, even with other therapies, the neurotox has appeared to be close to zero or much less.
Nice.
And again, is that something different in the target, or it's a different in the dose levels? I think we'll see. But I think we know a lot more on how to manage CRS in general. We have algorithms at each expert center. When a patient meets grade 1 for X hours or grade 2, you kind of do this or that. Now we still need to learn and keep an open mind that with AML CAR, that maybe we have to have a somewhat unique approach, but I think we've learned so much that we're better capable of managing those toxicities.
Okay. In CARs in general, I think there is a requirement for lymphodepletion. Why is that? And I think I heard you mention that in the UltraCARs, you use Cytoxan for that. And is that a requirement in the UltraCARs as well?
Yeah, so I think the goal, the standard sort of lymphodepletion is some combination of the drugs fludarabine and cyclophosphamide, ranging from anywhere between 2 days and 4 days in dosing, both across approved and not approved. The reason why you need it is that each patient has their own T cells, and their T cells unfortunately can reject these T cells, even though they're autologous. And so you're depleting their host T cells to allow these cells to best expand and persist. So every approved CAR-T therapy has some level of lymphodepletion with the combination of fludarabine and cyclophosphamide. Again, what total number of days and total dose, I think people are still seeing if there's a difference. It appears there doesn't really appear to be a difference. It's just important that you give it. And myeloid CAR, we have tried to not give it. So for example, even in our current trial, we have 2 cohorts. One gets no lymphodepletion, and they're basically 1 dose level above the cohort that gets lymphodepletion. The thought is that maybe with membrane-bound IL-15, could this overcome the need of the lymphodepletion? I have no idea if that is true or not true. And my kind of gut tells me that probably lymphodepletion will be ultimately required, but we are actually testing with and without. I think, a lot of this speaks to efficacy. Again, we've not had any any approved therapy without lymphodepletion. So just, common sense being likely it will be required.
Okay. And I think my last quote unquote prepared question is around CAR manufacturers. In the CAR-T for myeloma, for example, there are many CAR-T manufacturers. What is a product portfolio for UltraCAR-Ts? I know of that one company, Precigen, that is being used now, but are there others?
Yeah, I don't know of another company that has the same type of platform, sort of this rapid autologous turnaround. Again, there are lots of off-the-shelf allo companies involved in the space. We talked about some of those issues. There are also other approaches. So for example, like NK therapies, there was just a recent presentation by Fate where they have sort of a kind of like the most potent NK cell you can have, and they did report responses early on in their dose escalation phase of their trial in heavily relapsed refractory patients. So I don't know if there's a similar platform, but there are other cellular therapies, not even just traditional CAR-T. There are TCRs, there are NK therapies, there are gamma delta T cells, all of this is early on. And again, the bar is low. I mean, we're really looking, if we can have a 20, 30% complete remission rate that lasts for 6 months, then everybody's extremely excited. Obviously, our goal will be to improve that. And our goal is, to Dr. Jude's point, to ideally cure these patients. But really on, we're just very, very early on. But from a rapid autologous turnaround platform, I think this one is currently, quite unique. Again, with all the challenges for AML-directed CAR that we've kind of mentioned earlier.
Yeah, if you want to go back to the audience questions.
Okay, hey, thank you. Thanks for coming. Thanks for coming back there, Yalik. We appreciate that.
I wouldn't miss your talk, Gary.
You take care. AML patients enrolled might see their AML cancer reduce or even cure, but they might develop other cancers. At JP Morgan, Helen, I guess she is the CEO of Precigen, Helen Sebzir-Vari, I guess that's her name. I'm sorry for the mispronouncing. Mentioned that Precigen will be in 2022 launching a library approach, which I would like you to discuss at all, whereby the UltraCAR-T can produce multiple CAR-Ts targeting different cancers on a pick, mix, and redose basis as needed. The AML patient enrolled in cohort 1 might not make it to 2022. Is this library approach going to be used earlier on a compassionate use basis for these enrolled patients who would otherwise die?
Yeah, so I think very clearly no, unfortunately, to that question. I think there is a ton of logistics in coming up with these therapies for our patients. And of course, and I think rightfully so, we have to prove safety. I think this kind of speaks to the future, because I think 3 to 5 years from now, and I know that that's 2022, but realistically longer than that, at some point we're going to know what the truly best antigens are to target. And again, with this library type approach, can we, choose, target A and B, and that's the best one for all, or does this have to be very personalized? So again, one of our issues is there is no perfect marker in AML. And so potentially each patient needs their own CAR-T approach. And with a viral vector base, that would likely be an impossibility. But if you could create it to where we've proven safety with CARs A, B, C, D, E, and F, maybe you have a 3 or 4. I mean, that would still be a huge number. And then you have your patient enrolled and you choose the CAR based on their screening flow cytometry of their bone marrow blast. So you get their bone marrow and you say, hey, this one has the highest expression of marker A and D. I'm going to choose that, or marker whatever. So I think that adaptability and personalization, which may ultimately truly be required for AML patients, has the ability here. Of course, we'd love to say, hey, BCMA for all or CD19 for all. That's not realistic. Again, we may have to, take a step back and say, hey, we're going for a 30% response rate. But if we really want to individualize it, having a library approach would be amazing. I think the red tape and the logistics and the challenges is you have to prove safety of all of these different approaches, or if you're going to sequence them or combine them. But at least with this, it would, one, allow you to adapt it. If you hey, marker A and D is the best, fine, we can do that immediately. But if we really have to personalize it, we could do that in the setting of a trial. It would be a first in ever trial, but there is that potential. Again, I think some of this is hope, but I do think there is that opportunity there.
So would there be like a BCMA, CD30, 19, CD38, like you pick?
Yeah, it could be. Yeah, it could be easily a you pick, option. I think, the challenges in the field is once you have an effective therapy, everybody wants to somehow duplicate it. But I think thinking about it and moving it to the next step, again, when you understand, well, why do patients with BCMA CAR progress? Are they BCMA negative, or what is their, resistance, factors, and how could you then give some other, CAR-T targeting something else, or can you rescue the CAR-T cells that are there? I think there's a lot of unanswered questions. I think what's nice is we do have the technologies to answer a lot of these questions in the setting of the current clinical trials. Again, in AML, we just lack a lot of anything from a data perspective. But that is the hope, that you could really real world personalize this therapy to a much greater perspective.
That's great. Now, are you seeing evidence in CAR persistence on the initial cohorts, which were given a low dose, contributing to some level of efficacy?
Yeah, I mean, we really, as far as public data, just have the one patient, and the answer is, her blast continued to go down over time, with persistence at least at 6 months. But I would, look forward to our future presentations. I think those are key questions. What is the safety? What is the activity? And then from a PK translational perspective, how durable, are these cells over time? I think are really critical questions, and I hope to be able to, present these data in the not distant future.
Okay. And did you own a Tesla, don't you?
I wish. I've looked at them before.
Oh, no, because I got to tell you, you're an early adopter, I think if you lived in the 50s, you'd be saying, sure, they're going to put a man on the moon.
Yeah. Yeah, I think in AML clinical trial research, we have to be, very optimistic. And I think, it doesn't take a lot of patients to do well with any of these therapies for the fields to get extremely important. And again, when you have, 3, 6, 9 patients, you want to be careful, how handpicked were these patients, how durable are their responses, et cetera. But I think, in the future, again, when you start to see cohorts of 20, 30 patients with reproducible responses with any of these therapies, and that's when I would really be getting excited, because that's just never been shown to date in the relapse refractory group. And that's really at minimum. Now, if you see something better, then that's even more of a home run. But I think you have to be optimistic in this field.
I love people like you who look at the most difficult problems that can exist in the field that they're in and choose to go head on.
Thank you.
So thank you so much. Priya, do we have any other questions from the audience?
No, nothing pertaining to the subject that we're discussing. So I think we can wrap up for today.
Okay.
Dr. Sallman, thank you so much. That was a great session and a lot of information shared. Gary, Jack, and Elek, thanks for joining. Your questions are absolutely great. And please visit curetalks.com for details of our upcoming shows. And this show will be uploaded right away onto the website.
And as always, thank you again, Priya, for putting this whole thing together, and Yelik and Jack for being such great participants, providing such excellent questions.
Thank you, Jack.